Greetings. We are now going to start coverage of the autonomic nervous system, chapter 14. Now, when we talk about this is a system that is outside of our awareness, which basically means that in a lot of cases, we are not aware of how our blood pressure goes up, blood pressure goes down, movement of food through the gastrointestinal system, the pace and intensity of the cardiac muscles, or release of lipids from adipose tissue. That's what we're going to explore today. Ganglionic neurons of the autonomic nervous system, from this point on we're going to refer to it as the ANS, control visceral effectors. Most of this control is outside of our awareness. Ganglionic neurons control visceral effectors. When we talk about this can be preganglionic neurons, are visceral motor neurons whose cell bodies lie in the brain stem and the spinal cord. Many of these are part of our visceral reflex arcs. Axons of these neurons leave the CNS and synapse on ganglionic neurons. Autonomic ganglia, which are visceral motor neurons in peripheral ganglia. They will innervate and control visceral effectors such as cardiac muscle, smooth muscle, glands, and adipose tissue. When we take a look here, this is a pure rendition of sensory information going up into the brain, initiating a response that is, for the most part, somatic, meaning that we're aware of this, it's going to control skeletal muscle, etc. But when we look at the autonomic nervous system, we may have information coming up into the brain, or it may be initiated from other stimulus, excuse me, and what we have is the autonomic ganglia being given specific instructions by the autonomic nuclei in the brain stem or spinal cord, and will initiate actions. Some of them will have to go through preganglionic neuron and then from there to a ganglionic neuron, and they will affect smooth muscles such as those in the urinary system, respiratory system, the walls of the blood vessels, the digestive system, etc., the glands, salivary glands, pancreatic, the pancreas, other types of glands that exist in the digestive system, etc., or the adrenal glands. They may affect the cardiac function, excuse me, and we're talking about cardiac muscle tissue, or the adipocytes, those cells that really store a lot of lipids and may have to be releasing it. Now, when we look at the autonomic nervous system, it consists of a sympathetic and parasympathetic divisions. Sympathetic is best known as the fight or flight. Kicks in during exertion, stress, or emergency. The sympathetic divisions, otherwise known as the thoracolumbar division, these axons emerge from the spinal cord, lateral gray horns of the thoracic and superior lumbar segments. The axons innervate ganglia close to the spinal cord. Whereas the parasympathetic, this is the rest and digest. It is predominant under resting conditions. The parasympathetic divisions, otherwise known as the craniosacral division, emerge from the brainstem nuclei and lateral gray horns of the sacral segments of the spinal cord. The axons innervate ganglia usually very close to the target organs. We have to keep mindful of one other particular division of the nervous system, that is the enteric nervous system, ENS. Excuse me. This is an extensive network of neurons and neural networks that innervate the walls of the digestive tract. We will go through more of this once we get to the digestive system in A&P 2. Here you see the sympathetic division, where most of the nerves are coming out in the thoracic to the upper lumbar. They will extend downward, and they will extend superior, excuse me, as a sympathetic chain ganglia. The parasympathetic, interestingly enough, come out from 4 cranial nerves. Number 3, 7, 9, and 10. 10 being vagus nerve. And then also from sacral nerves, this is coming out from S2, S3, and S4. What you see here in this image is really an example of the enteric nervous system. You can actually see some of the ganglia there. And these are really critical for the timing, the proper secretions, the proper functions of a lot of the digestive system. That doesn't mean that the autonomic nervous system doesn't play a role in it, but that really it's been a discovery in the past 50 years now that the enteric nervous system has been playing more and more of a key role. And this is where we get into the brain-gut connection. And that is when they were hunting for, when scientists were hunting for neurotransmitters, what they found in some case was a unique neurotransmitter, for example, in the gut. And usually the sense was, well, if you find it in the gut, you're going to find it in the brain. And they did. Consequently, if they found it in the brain, they would then look for it as a neurotransmitter somewhere in the GI system, and they would find so. Now, if we take a look at this gentleman here, we have a variety of plexuses and ganglia that you will see. Some of them are going to play a key role in AMS processing of information, either parasympathetic or sympathetic. But you need to be aware of that the variety of plexus may contain some of the nerves involved in the AMS system. The sympathetic division has chain ganglia, collateral ganglia, and then also the adrenal medulla. Now, the parasympathetic division has terminal, or what is referred to as intramural ganglia. When I talk about sympathetic, you will have preganglionic neurons. They will meet, and then you will have ganglionic neurons, and then sympathetic chain ganglia. Collateral ganglia, which include the celiac, the superior mesenteric, and the inferior mesenteric. And then a unique situation occurs with the adrenal medulla, which I'll explain a little bit more in detail in a few minutes. The parasympathetic consists of the cranial nerves 3, 7, 9, and 10. And you will also see them coming out from the lateral gray horns in the sacral region from S2, S3, and S4. You will have the presence of ganglionic neurons. And you will also have an unusual situation of the ganglia coming out from the ciliary, pterygopalatine, submandibular, and optic ganglia. There will also be some intramural ganglia from basically cranial nerve 10, which is the vagus nerve, as well as pelvic nerves. You want to review the targets of where they're going to be at, and we'll get more into that in a few minutes. The general pattern of sympathetic activity is a heightened mental alertness, increased metabolic rate, decreased digestive and urinary functions, activation of energy reserves, dilation of respiratory passageways, and increased respiratory rate. Interestingly enough, also at the same time, you're going to have an increased heart rate and blood pressure, and activation of the sweat glands. To give it to you in a sort of a nutshell, think about it this way. As I said before, fight or flight. What you have to do is sequester a lot of the blood away from non-essential functions, non-essential for immediate survival. And so you're going to see a lot less of the blood going to GI, urinary, and a few other places, and a speed up of oxygen-rich blood by the increased respiratory rate, and an increase in blood flow, not only to the skeletal muscles, but the cardiac muscle as well. You will also have activation of the sweat glands for thermal regulation, an increase in the metabolic rate, and a heightened mental alertness because of the threats or emergency or whatever. The general pattern of parasympathetic is the opposite. You have a decreased metabolic rate, decreased heart rate and blood pressure, increased secretion of salivary and digestive glands, increased motility and blood flow in the digestive tract, stimulation of urination and defecation. This is rest, digest. Some people would call it rest, digest, and defecation, but here's the key point here. The blood flow is now not necessary for emergencies, survival needs, etc. Rather, we are going to be allowing more of the flow to occur through digestive system, through elimination of bodily waste, such as through the production of urine and also defecation activity. You're going to have also the digestive tract becoming much more active to pass waste as well as to process the food products to obtain nutrients. This is where you're going to have more blood flow into areas such as intestine, to the liver, to the pancreas, etc. More secretion from a variety of glands that are involved with the digestive process, so that salivary glands, the stomach acid and stomach enzyme, the pancreatic, the liver, production of bile, etc. And I can go on and on and on, but basically this is what is going on. Now, when you look at preganglionic neurons, think about it this way. If one was to talk about the somatic nervous system, that's basically where you're having conscious control of skeletal muscles. You don't need multiple layers of neurons. You have one neuron leave the spinal cord and have its motor end plate connected to a muscle. Here, though, we have several routes possible. Now, in this case, what we're going to be dealing with, this is involving, first and foremost, the sympathetic nervous system. So I'm going to have preganglionic neurons leaving the lateral gray horn. Okay, remember, the lateral is only showing up in the thoracic and the lumbar, where basically you have, for the most part, the need for the sympathetic activation, or the autonomic nervous system, I should say. First, you could have this attached to the sympathetic chain ganglia, which is really a chain of ganglia that is outside of the actual spinal cord itself. These will basically synapse. These neurons will travel from that chain ganglion, and their postganglionic axons will eventually connect up with visceral effectors of the thoracic cavity, head, body wall, limbs. Collateral ganglia, these are the ones that are going to have, in some cases, clusters of ganglia outside and a distance away from the spinal cord. So the preganglionic neurons travel further and farther until they reach these ganglia, and the postganglionic neurons will travel and affect the visceral effectors of the abdominal pelvic cavity. So that would be things like the digestive system, urinary system, etc. Now, there's an interesting quirk here I want you to be aware of. Preganglionic neurons leaving the spinal cord, but going straight over, and this preganglionic will actually innervate the adrenal medulla. Now, in the adrenal medulla, you have to understand something. This is a completely different case. We're not talking the exterior cortex, but we're talking the inner core. And in the inner core are these cells, neurochromaffin cells. These chromaffin cells release epinephrine and norepinephrine. The interesting thing is scientists have found if you remove those cells and put them into a culture dish under the right conditions, guess what? They'll sprout axons. It's almost like they were nerve cells from the very beginning, but now they've been kind of encapsulated inside of an adrenal gland. Here's the main point also. The autonomic nervous system has several routes to activate the fight or flight response. And one of them will be, interestingly enough, this release of epinephrine and norepinephrine into the blood, which will enhance some of the fight or flight responses, and it will last a little bit longer. Thereby, it enhances the survival of the organism. Now, if you take a look at the parasympathetic, it's a little bit different. Here you have basically 4 cranial nerves. Okay. Cranial number 2 will basically have its preganglionic neurons attach and come in contact with the ciliary ganglia. Their secondary, or excuse me, postganglionic neurons will then match up and in this case affect the intrinsic eye muscles, thereby altering pupil and lens shape. Cranial nerve number 7 will meet up with the pterygopalatine and the submandibular ganglia. That's where the preganglionics meet the postganglionic. The postganglionics will send out their axons and they will connect up with nasal glands, tear glands, and salivary glands. Cranial nerve number 9, its preganglionic will eventually meet up with the optic ganglia, and its postganglionic neurons will synapse or act on the parotid salivary glands. Finally, cranial nerve number 10. This is the one that reaches farthest. It's the vagus nerve. And this is going to basically synapse with intramural ganglia. These, in other words, are short distance ganglia. You have very, very long preganglionic axons that are going to stretch out. These ganglia are going to be very close to the visceral organs of neck, thoracic cavity, and most of the abdominal cavity. Okay, so a lot of the organs in thoracic cavity, heart, lungs, abdominal cavity, stomach, small intestine, large intestine, liver, gallbladder, pancreas, things like that. Now, also notice down at the bottom, coming out of the lateral gray horns of the sacral spinal cord segments, S2, S3, S4, you will have preganglionic fibers. They're going to be the pelvic nerves. They are going to synapse at ganglionic neurons that are in the intramural ganglia. If you get confused with intramural, think of it as really close up to the target organ. In this case, where they're going to synapse is the visceral organs in the inferior portion of the abdominal pelvic cavity. Think of it as the more terminal or lower in the abdomen area of sex organs, the large intestine, the bladder, okay? All of that. Now, the AMS divisions innervate many of the same structures, but the innervation patterns are very different. These are the notes or concepts you really got to get. Every spinal nerve has a gray ramus, communicans, and that it carries a sympathetic postganglionic fibers to the body wall and limbs, okay? Excuse me. The superior cervical sympathetic ganglia supply regions innervated by cranial nerves 3, 7, 9, and 10. Splanchnic nerves supply the viscera. Okay, so what we're talking about here is cardiopulmonary, postganglionic innervation of heart and lungs, abdominal pelvic. This is the preganglionic fibrous synapse in the collateral ganglia of the celiac, superior and inferior mesenteric, and the pelvic, which is the postganglionic fibers inferior to L2, innervate the pelvic cavity. And as I mentioned before, the adrenal gland is postganglionic cells. Wow. So what you're seeing here is this. Here you have, and you know that this is all sympathetic because you start from T1, you end up at L2. In a lot of cases, some of these pathways, here is the spinal ganglionic chain, okay? And you have pathways that will synapse at this ganglia, and so the fibers that leave are postganglionic. Notice that they come together in certain plexi and eventually go to the certain specific target organs, the heart, the lung, in other cases, the salivary glands and the eye. But other cases, the preganglionic will go beyond the point of the ganglionic chain, go to other ganglions, such as the superior mesenteric one, the inferior mesenteric, and the celiac, and then have their post synaptic neurons follow down and then innervate liver, gallbladder, stomach, spleen, pancreas, small intestine, large intestine. Now notice also that there is this nice little plexi right here, and this is moving out from sort of a lower end of that chain. Don't get it mixed up with the sacral part later on that's far down deeper that really is involved with parasympathetic. This is all sympathetic now. Two things to keep in mind. This lower chain will reach out and have postsynaptic connections with ovary, uterus, penis, scrotum, urinary bladder, kidney. Okay, remember that adrenal medulla? That's a very special case there. And then the other thing I wanted to bring up to your attention is this: the postganglionic fibers to the spinal nerves that innervate, surprise, surprise, the skin. What I mean by that is blood vessels, sweat glands, erector pili, and the adipose tissue. This is going to be important also in being able to function, achieve homeostasis under conditions of sympathetic stimulation. Now, AMS innervates many of the same structures, but the innervation patterns are different. This is when I mention this, you have to keep this in mind that there are dual innervations with certain organs. Let me go up to the parasympathetic notes. The vagus nerve provides roughly 75% of all parasympathetic outflow. Nerve fibers do not join the anterior roots of the spinal nerves. The preganglionic fibers form pelvic nerves that innervate intramural ganglia in the walls of the kidneys, urinary bladder, terminal portions of the large intestines, and sex organs. What I'm saying there is you end up having this much farther length of a preganglionic fiber in parasympathetic. Usually, the pathway from the ganglia and the postganglionic fiber, they're very short because they're really right up against those organs that they're going to innervate. You want to note the ganglions that do exist: ciliary from cranial nerve 3, pterygopalatine from cranial nerve number 7, submandibular from cranial nerve 7, and the octic ganglia from cranial nerve number 9. And you can see them right here. You'll note, interestingly enough, that the facial nerve, which is really number 7, has a collaterals that innervate the pterygopalatine ganglia, which innervates the lacrimal gland. It also innervates the submandibular ganglia, which innervates really some of the salivary glands right here. So gland here, but they're very different. Also note the ciliary ganglia right here. If you think of ciliary, think of the ciliary muscle that controls the focus of the lens in the eye, as well as influencing the iris so that it controls the amount of light that comes in. It gives you a focus, okay? Notice you have a cardiac plexus here, which innervates both the heart and the lungs. Again, this is parasympathetic, not sympathetic. You have a celiac plexi here, which will then have innervations of liver, gallbladder, stomach, spleen, pancreas, etc. An inferior mesenteric plexi, which eventually gets into small and large intestine. A hypogastric below the stomach. So you end up having these innervations here, along with some of the pelvic nerves. You finally get into the reproductive organs, urinary bladder, and the kidney. Parasympathetic does not innervate the adrenal. Keep that in mind. The functional differences between these autonomic divisions reflect their divergent anatomical and physiological characteristics. Again, this is basically what you're going to start seeing a lot of. Sympathetic, fight or flight. Parasympathetic, rest and digest. But not only do you see it by innervation, but you see it also by the neurotransmitters involved. Okay, let's go through this. When we talk about the sympathetic, the preganglionic neurons all have, as a neurotransmitter, acetylcholine. They're known as cholinergic neurons. Postganglionic neurons will discharge, they're known more as catecholamine or catechols. They release norepinephrine and epinephrine. Now, Norepinephrine will be the neurotransmitter of choice for postganglionic neurons of sympathetic. You get norepinephrine and epinephrine from the adrenal medulla, because that's a different little animal of itself. That's the exception to the rule. You have a few unique situations of acetylcholine still being used, and that happens with the skin. Now, in sympathetic activation, the entire physiological system is activated. It's controlled by the sympathetic centers in the hypothalamus. What about the parasympathetic? Well, preganglionic neurons use acetylcholine, so they're cholinergic. Postganglionic neurons use acetylcholine. They're cholinergic. This is more referred to as the anabolic system for the parasympathetic because it's used to build up nutrient content in the blood. There are some effects of sympathetic activation you need to be aware of. You're always going to get this sense of increased alertness, people feeling on edge, a feeling of energy euphoria, increased activity of cardiovascular and respiratory centers in the pons, medulla oblongata. This is going to increase blood pressure, heart rate, breathing rate, and depth of respiration. An general increase in muscle tone. You're going to see this through the stimulation of the medial and lateral pathways, so the person looks tense and may even begin to shiver. Mobilization of energy reserves. This is going to be through the accelerated breakdown of glycogen in muscle. Now, you're going to see the accelerated breakdown of glycogen in liver cells to release more glucose as well. And that's going to be particularly through norepinephrine and epinephrine that's been released into the blood from the adrenal gland, and that's going to have a little bit longer time to provide fuel. You're also going to get more fuel for the cells by the breakdown of lipids and release of these lipids from the adipose tissues into the blood. So basically, when you look at sympathetic, you have a preganglionic neuron in the CNS. It will then synapse here in the sympathetic ganglion, and then its postganglionic fiber will eventually send out, and it will release norepinephrine as a neurotransmitter to the target organ. Now, if you end up with your target being the adrenal medulla, you don't have any postganglionic fiber. You've got the medullary cells in there. They release immediately epinephrine, norepinephrine right into the bloodstream, and that has a prolonged effect during sympathetic activation. Okay? Well, what about parasympathetic? Preganglionic, it's going to be in the CNS. Preganglionic fiber goes into the PNS. Preganglionic basically will synapse with the ganglionic neuron. Notice the length of the postganglionic fiber, very short. A lot of these ganglion or a lot of these fibers are right next to the target. So it's the preganglionic fibers in the PNS that are usually very long, and they always are just using acetylcholine. Effects of parasympathetic activation. Okay, here's the grocery list that you really need to start thinking about. With sympathetic, you get a dilation of the pupils to increase the amount of viewing, although you do not have fine detail vision. Opposite occurs with parasympathetic. Constriction of pupils, you restrict the amount of light, and you also have more of a curvature of the lens of the eye. That gives you view of nearby objects. You will have an increased secretion of digestive glands, including the salivary glands, gastric glands, duodenal glands, intestinal glands, pancreas, the exocrine and endocrine, and the liver. Secretion of hormones that promote the absorption and utilization of nutrients by peripheral cells. Changes in blood flow and glandular activity associated with sexual arousal. Increased smooth muscle activity along the digestive tract, stimulation, coordination, defecation. Contraction of the urinary bladder during urination, constriction of the respiratory passageways, reduction of heart rate and focus of contraction. Force of contraction. Now, membrane receptors at target organs mediate the effects of the sympathetic and parasympathetic stimulation. It's not Medicaid. Spell checker has to be punished later. When we talk about neurotransmitter release for the sympathetic, we have really the adrenergic has two sets, two classes, and each class has several subclasses. So let me go over this with you. With adrenergic, you're going to have alpha receptors, alpha 1, alpha 2. They're all G protein activation of the cells. Beta receptors are beta 1, beta 2, beta 3. They're all located on cell membranes of the lungs, heart, liver, and other organs. They, again, are G protein activation. This triggers changes in the metabolism of the cell. Activation of different receptors may trigger different activities in the target cell. Norepinephrine stimulates alpha receptors to a greater degree than beta receptors, whereas epinephrine stimulates both beta and alpha classes. Norepinephrine release from postganglionic neurons. Okay. Norepinephrine and epinephrine are released from the adrenal gland. Here's the ratio. Epinephrine 80%, norepinephrine 20%. Now there's an advantage here also because epinephrine will linger longer and the beta effects seem to predominate. The adrenal gland chromaffin cells act as postsynaptic ganglionic cells, and that's where I brought up that point about neural in origin. I included this from another text to help you to review this. When we talk about acetylcholine, we're going to be talking about cholinergic receptors. There's only two major types, nicotine and muscarinic, or nicotinic and muscarinic. When you get to adrenergic, though, beta 1, beta 2, beta 3. The beta 3 is only unique for adipose tissue. Beta 1 is predominant on the heart. Beta 2 you'll see with lungs. It's abundant in blood vessels serving the heart, liver, and skeletal muscle. So really what you want us to do is know the major locations? Yes. And these receptors. Alpha is most important in blood vessels serving the skin, mucosa, abdominal viscera, kidneys, salivary glands, also virtually all sympathetic target organs except the heart. Alpha 2, the membrane of adrenergic axon terminals, pancreas, and blood platelets. You see, you're going to notice one of the things right down here. When you have sympathetic activation, fight or flight, one of the things that activation also does is it promotes blood clotting. Well, if you're going to fight something, you want to have an advantage, and the advantage is reduction in blood loss by blood clotting better. Now, I bring up this is one of these simple charts that shows you norepinephrine binds to an alpha receptor, activates a G protein, and if it's alpha 1, you go through secondary messengers, the release of calcium from the ER, and these are the results. Smooth muscle contraction, glandular cell secretion. If it's alpha 2, you have reduced cyclic AMP levels and an inhibition of the cell. If you have epinephrine and it binds to the beta receptor, you activate the G protein, activation of adenyl cyclase. This is going to take and make cyclic AMP. And if cyclic AMP is made, then it depends on whether you have beta 1, beta 2, or beta 3. Beta 3 is the biggie when it comes to dealing with, excuse me, fatty acids and adipose tissue. Okay? Now, membrane receptors at target organs mediate the effects of the sympathetic and parasympathetic stimulation when it comes to cholinergic. Remember in here, we're dealing with the parasympathetic, and they only use acetylcholine for both pre-ganglionic and post-ganglionic. Now, there's something that you have to keep in mind. We have nicotinic receptors and muscarinic. Both of these were found by examining how two different types of poisons, that's what nicotine is, acts. And actually, nicotine was used as a insect pesticide. Yep, they used it then. Now, of course, everybody frowns on it because, well, it's an addictive drug, and people get it with smoking and all that stuff. But just take this from the basic knowledge that was first found: the neurochemistry of these receptors. A nicotinic receptor is an open ligand-gated ion channel. That's it. Nicotine binds to it, acetylcholine binds to it. found in all postganglionic neurons, excuse me, adrenal medullary cells, and the neuromuscular junctions of the skeletal muscle cells. It's called nicotine from the effects of nicotine. Now, muscarine is from a poisonous mushroom. It activates a G protein-coupled receptors, occurs in cholinergic neuromuscular or neuroglandular parasympathetic division. You'll find those in the cells, and a few cholinergic junctions in the sympathetic division. I want you to be mindful of one other thing too. G protein activation tends to last longer than ion-gated channel effects. What does that mean? Simple. If I activate this, whether it's by injecting acetylcholine, being exposed to nicotine, being exposed to muscarine, when I'm dealing with G protein, it's going to take a little bit longer to make the connection inside the cell and changing things. But its effects are going to be much longer time-wise versus turning on and turning off a ligand-gated ion channel. Now, one other thing to keep in mind. You're going to have, if you have a G protein cascading event going on inside the cell, you're going to have activation and inactivation of certain enzymes inside that cell that may act in an inhibitory or excitatory effect. Here's the cytoplasm of a target cell. Here's our nicotine receptor. You see the choline binds to it, opens up a sodium ion gate. Fine. Here is acetylcholine attached to a muscarinic receptor, activating the G protein. And suddenly now you've got activation and inactivation of all sorts of enzymes. It has a cascading effect inside the cell, which will alter metabolic effects on the cell. Now let's step back for a second and think about this for a minute. The autonomic nervous system adjusts visceral motor responses to maintain homeostasis. Now keep in mind, visceral motor is not skeletal muscle. We're talking about heart. We're talking about smooth muscle. We're talking about glandular secretions, etc. The physiological responses are maintained independent of consciousness or conscious control. Translated, folks, as I've said it a bunch of times before, you're sitting there and listening in a lecture. You're not telling your heart, okay, beat again, breathe, heart beat again, breathe. Okay, now dump from the stomach into the small intestine. You don't think about any of those. Those are autonomic. They're automatic. They're just happening. But it is the ANS that is absolutely critical to maintain homeostasis, especially for digestive, cardiovascular, respiratory, and reproductive systems. We've seen this chart before. We start off with a stimulus. This is picked up by general sensory receptors, passed up sensory pathways to the sensory processing centers in the central nervous system. Now, motor centers are operating at the subconscious control, and there's a back and forth that occurs. The processing may be at the conscious level, which can be affected by memory, learning, or planning. But what do we have? We have motor responses. Now, the motor pathways are somatic. That's the somatic nervous system, which, of course, when we talk about somatic effectors, is your skeletal muscles. Okay, but let's get back to where the main topic is for this chapter, and that is autonomic nervous system. You're not conscious of that. You don't have to be. And it affects visceral effectors, smooth muscles, cardiac muscle, adipocytes, glands. The ANS provides precise control over visceral functions. The following tables are used for comparisons of sympathetic and parasympathetic divisions. But I want you to note that sympathetic has more tissues and organ targets than parasympathetic. You will see many examples of dual innervation. In other words, that's basically innervation of both sympathetic and parasympathetic, and they have opposing effects. But not all of them have dual innervation. Here's what I mean. If you take a look at sweat glands, erector pili, yep, you got innervations from sympathetic effects, and it tells you what's going on here. And the nice thing is it tells you what are the alpha or beta subtypes that are playing a role. But notice, in the skin, no parasympathetic effects. Okay, let's go over to the eye for a second and look at this. Sympathetic effect, dilation of the pupil, accommodation for distance vision. Parasympathetic, constriction of the pupil, accommodation for close vision. If you look at the blood vessels in the cardiovascular system, just for example, you have dual innervation only at the heart. In the veins, nope. In the blood vessels to brain, kidney, digestive viscera, lungs, heart, skeletal muscle, skin, nope. You do have sympathetic innervation to skin, skeletal muscles, heart, lungs, digestive viscera, kidneys, and brain. You'll notice that a lot of it is dilation to the critical structures that are necessary for fight or flight, not dilation, constriction actually, to digestive viscera. And in a fight or flight situation, you will end up with constriction, reduction in blood flow to the kidneys, so that means reduction in urine production. Let's take a look over here. Adrenal gland, sympathetic effects, secretion of epinephrine, norepinephrine. Parasympathetic, nope. Neural hypophysis, nope. Secretion of ADH. Pancreas, decreased insulin secretion. That's for the sympathetic. For the parasympathetic, increased insulin secretion. Pineal gland, increased melatonin secretion. Parasympathetic, inhibition of melatonin synthesis. Now, dual innervation exists with certain structures to increase or decrease what's going on. If you always get lost, go back to the beginning. Sympathetic, fight or flight. Parasympathetic, rest and digest. These here in the digestive system, salivary, sphincters, general level of activity, secretory glands, liver, pancreas, all of these are going to be having an advantage for rest and digest. At the same time, under sympathetic, there's going to be a reduction in a lot of this. The only thing you're going to see really is a revving up of the release of glucose because it's needed for fight or flight. When we get to the adipose, there is no parasympathetic effect. There is a sympathetic effect, lipolysis, meaning the breakdown of fats, release into the blood. Okay? So these are the things I want you to keep in mind. Now, dual innervation also does play a role in a sort of synergistic approach, and this was found with the reproductive system. In sympathetic, it seems as if the sympathetic and parasympathetic play a role in certain steps during human reproduction. Male, increased glandular secretion, ejaculation for sympathetic, but in the parasympathetic, first you have the erection. For the female reproduction, you're going to have variable depending on hormones present. That's for parasympathetic, but for the sympathetic, a relaxation of non-pregnant uterus and increased glandular secretion, contraction of pregnant uterus. As you can see here, this diagram shows very nicely heartbeats per minute. Heart is one of the best examples of dual innervation. So when you have an increase in parasympathetic stimulation, you have a decrease of the heart rate. When you have parasympathetic inhibition or sympathetic stimulation, this increases the heart rate. If you have basically the and, where you have sympathetic stimulation combined and with parasympathetic inhibition, you're going to have a maximum increase in the heart. And that's where Lowy was able to do his work with stimulating the nerves that both innervated the heart from the parasympathetic, and one was from the sympathetic. Now, most visceral functions are controlled by visceral reflexes. Visceral reflexes are autonomic reflexes initiated in the viscera, the heart muscle, glands, digestive system, etc. These reflexes provide automatic motor reflexes that can be modified, facilitated, or inhibited by higher centers, particularly by the hypothalamus. Remember, the hypothalamus is kind of like one of the key players in sympathetic and parasympathetic in ANS. That's one of the control areas. Keep in mind that all visceral reflexes are polysynaptic. We've talked about polysynaptic versus monosynaptic reflexes before. The components of a visceral reflex include the receptor, the sensory neuron, the processing center, which is going to be one or more interneurons, or two visceral motor neurons. Most visceral functions are controlled by visceral reflexes. If you have a short reflex, they may bypass the CNS entirely. The control activity in one part of the target organ, and they predominate in the enteric nervous system. So going over that, it's short, it doesn't have to have the CNS involved, and it only affects one part of the target organ. You see a lot of it in the ENS. For long reflexes, they deliver information to the CNS along with posterior spinal nerves, within the sensory branches of the cranial nerves, within autonomic nerves that innervate the visceral effectors. They control the activity of the entire target organ. Now, the visceral sensory information is delivered to the solitary nucleus. Remember, that's the nucleus I didn't spend a lot of time in the last chapter, but it's located in the medulla oblongata. Almost all processing of sensory info is done at the subconscious level. And you see that right here. Stimulus, receptors in the peripheral tissue. This is an afferent pathway. It goes up. You have an autonomic ganglion. You have an interneuron here. It's polysynaptic because there's 1, 2, and finally the third synapse here to the peripheral effector. So it's a very short reflex. If you take a look at the long reflex, you have your afferent fiber, which is a sensory fiber, comes in, gets right into the CNS. So you've got one synapse here. You've got a association or processing center neuron, a CNS neuron here, interneuron. So you've got one synapse, two synapses, three synapses, and the fourth one. So what's going to happen here is this information is going to pass out on this preganglionic neuron, come up to an autonomic ganglia, have a postganglionic neuron, and eventually to its peripheral effector. We need to bring up interoceptors, which provide visceral sensory information. Now look, this is where we're dealing with either cranial nerves, the posterior roots of spinal nerves, T1 to L2, so that's what? Sympathetic. Cranial nerves are going to be parasympathetic. The posterior roots of spinal nerves, S2 to S4, that's parasympathetic. So what are we getting this sensory information from? Well, let's see, mouth, palate, pharynx, larynx, trachea, esophagus, associated vessels and glands. So a lot of that right off the bat is respiratory system, mouth, etc. When we talk about sympathetic, this is visceral organs located between the diaphragm and the pelvic cavity. Gosh, that sounds like most of the digestive organs. And finally, the posterior roots of the spinal nerves, the organs in the inferior portion of the pelvic cavity, that again is parasympathetic, okay? And all of this is going to get processed and delivered to the solitary nucleus, okay? So, no, you won't have to memorize this whole chart. Don't lose it, okay? There are a variety of things you could go over on this because you see that there's a lot of sympathetic reflexes, but even more parasympathetic. Coughing, swallowing, consensual light response, urination, defecation, gastric intestinal reflexes, baroreceptor reflex, sexual arousal. Meanwhile, on the sympathetic side, we've got cardioacceleratory reflex, vasomotor reflex, pupil light reflex, and ejaculation in males. Well, here's the comments. It's just to give you a general idea that these reflexes exist. They reflect both sympathetic and parasympathetic. And yet they interact with either the ENS or the CNS in different ways. Let me come back to an area we talked about earlier, baroreceptors and chemoreceptors. They initiate important autonomic reflexes involving visceral sensory pathways. To help you to understand this, keep in mind this. These are internal sensors, okay? They are monitoring what is absolutely essential for certain biological functions, blood pressure. We're going to get into chemoreceptors in another minute. We're talking about dissolved oxygen, dissolved carbon dioxide, pH of the blood, okay? So these would be the sensory areas and the sensory information picked up by these particular key receptors. A baroreceptor is a stretch receptor that monitors changes in pressure. During pressure changes, the walls undergo changes in stretch or recoil. Okay, so what are they? They consist of free nerve endings. They branch within the elastic tissues in the walls of hollow organs, intestine, blood vessels, particularly huge elastic arteries. You're going to see this tubes of the respiratory, digestive, and urinary tract. Okay, fine. So maybe in the bronchioles or in the trachea, in the esophagus, in the stomach, in the ureters, in the bladder. Now, baroreceptors monitor blood pressure in the major vessels. You have the carotid sinus and the aorta, otherwise known as the aortic sinus. Here you can see some of this. So basically, as your aorta stretches, you're going to get certain pressure information. Now, you also have up here these structures called the carotid sinuses. They're going to monitor the blood pressure and alter by reflexes what you may need at the time by passing that information up to the brain and then downward to affect things like stretching, reducing the pressure, and you reduce the stretch of the aorta walls or the carotid sinuses, and so the pressure drops. So how would you do that? You could relax muscles, the smooth muscles in some of the arteries, but the other way to do it would be just basically to reduce the heart rate or the volume that's being pumped up by the heart per beat, okay? You notice that there are other baroreceptors there of the colon, bladder wall, digestive tract, lungs, etc. Now, what about chemoreceptors? These are very specialized receptors. They detect small changes in concentration of specific chemicals or compounds. So where are they located? The medulla oblongata. Some of them are also in other places in the brain, in the carotid bodies, near the origin of the internal carotid arteries on each side of the neck, in the aortic bodies, between the major arches of the aortic arch. Now, carotid and aortic bodies are small masses of tissue. You're going to just see them, they're just a little bit. They have receptors, they have supporting cells. So what are they checking? The medulla oblongata is basically involved with the respiratory center. It's going to monitor the CSF, cerebral spinal fluid, for pH changes and PCO2. When you say something like P, it refers to partial pressure. So not the pH, but basically the PO2 and the PCO2. This is the partial pressure of the gases present. So what happens is that when you have a buildup of carbon dioxide, the blood is also going to have a pH drop. So that's why the CSF is being monitored for pH changes. Now, in the aortic and the carotid bodies, you really directly measure the blood for both pH, PO2, and PCO2. And you can see these here. Now, this is extremely important because when these begin ringing their bells, if I could say it this way, you're going to see some changes because you have to start making correction. Blood is going to have to get moved faster. Oxygen has to get in there and you have to reoxygenate the blood. That is if you have a buildup of PCO2 or a drop in the pH. Okay. And this is how the cells will respond and the body will respond. And you're basically, this is again an issue of how you maintain homeostasis. Now, there are multiple levels of motor control for the ANS. The key one is the hypothalamus. It is really has several nuclei there that are control the sympathetic and the parasympathetic divisions. Next would be the medulla oblongata, which contains centers for cardiac, vasomotor, swallowing, coughing, and respiratory. More complex sympathetic and parasympathetic reflexes are really coordinated by these processing centers in the medulla oblongata. Now, the hypothalamus is also going to interact with other portions of the brain. Activity of the limbic system, thalamus, and the cerebral cortex can have significant effects on autonomic function. Basically, you can have other systems that will kick in if it is necessary, or if the hypothalamus is not functioning properly. But think about it this way. If I'm having problem breathing and I'm not getting enough oxygen and my PCO2 starts rising and my pH in both my CSF and my blood are dropping, what else is going to happen? Well, I'm going to try to increase my respiration rate. Now, some of those muscles that are involved with respiration are what? Somatic nervous system controlled, like the diaphragm and the external intercostals and the internal intercostals. So some of this information may get passed over to the hypothalamus, and so you have the cerebral cortex, and it's going to start working to really increase activity of respiration so that the PCO2 starts dropping, the PO2 increases, and the pH begins returning back to normal levels. And that's what you can see in this example here. Okay. So here we go back to another situation that we talked about earlier. We have information that gets passed up through sensory receptors, sensory pathways. That's our stimulus. The somatic reflexes, pretty straightforward. Somatic nervous system, skeletal muscles, okay, fine. Information gets passed, relayed, and processed centers in the brainstem. The visceral sensory gets passed to the hypothalamus. The somatosensory goes from the thalamus up to the cerebral cortex. There may be from the cerebral cortex motor commands back down through the SNS to the skeletal muscles. There's going to be some considerable chatter between the cerebral cortex, the limbic system. Notice that the arrows are both back and forth, back and forth. You're going to have communication between limbic system, thalamus, and hypothalamus. And eventually, you're going to have your visceral motor responses. Some of them may be long reflexes, and they're going to go through the autonomic nervous system. Some of them may be short reflexes. Notice that the pathway between here to here is very short, simply sensory to ganglionic neuron to visceral effector. Long has to go through the sensory pathway and has to go back down. But no matter what you do, you still have the visceral effectors affected. Wow, that was a quick chapter. And I appreciate your patience on this. I encourage you to peruse chapter review, pages 539 to 540. Complete your review questions on pages 541 to 542. Complete review sheets on pages 528 and 538. And the next chapter is the special senses. Thank you for your time.